Hai semua, in today's lesson, we will look at the chromosomal mutation and we will focus on the change in the chromosomal structure. So what do we mean by the chromosomal mutation? It is a change that occurs in the structure or the number of the chromosome. For example, we can see here that the structure of the chromosome changes as a part or a segment of the chromosome is being deleted or removed. And if we look at this diagram down here, this will be showing a karyotype of the human chromosomes. And we should know that the chromosomes of the humans will be 46. And if we really look at this karyotype, we will be able to see that there will be an abnormal number of chromosomes in which there will be 3 chromosomes number 21. And so this will be showing the change in the number of the human chromosomes. And so there will be 2 types of chromosomal mutation. One will be about the change in the chromosomal structure, like this one. And the second type will be the change in the chromosomal number, like the one shown in this karyotype. We will now focus on the change in the chromosomal structure, in which there are 4 types of chromosomal structure change that we have to know. Number 1 will be deletion. Number 2, inversion. Number 3, translocation. And number 4 will be duplication. We will start with the deletion. This is the loss of a segment containing 1 or more genes, as seen in this picture. And we can see that 1 segment of the chromosome is being deleted. Thus, finally, the structure of the chromosome changes in which it becomes shorter. How can this happen? So let's say we have 1 chromosome over here containing genes A, B, C, D, E, F, G, H, I. And let's say part of the segment breaks off. Okay, let's say over here and over here. And when this segment over here breaks off, it will be removed from the chromosome, and these 2 parts of the original chromosome will be joined with each other. Thus, this will create a new structure for the chromosome, which is shorter, and the genes that are found in this new chromosome will just be the genes A, B, C, G, H, and I. One example of a genetic disorder caused by deletion is a syndrome known as the Cri-du-chat syndrome or the cry of the cat syndrome. This is due to the deletion of the short arm of the chromosome number 5. If you refer to this diagram, we can see that this will be the chromosome number 5 of the humans, and in this case, a segment of the short arm for this chromosome number 5 has been deleted. Individuals having this Cri-du-chat syndrome is usually showing a distinctive cat-like cry and with some facial growth and mental abnormalities. The second type will be inversion, in which a region of chromosome breaks off and then rotates 180 degrees before rejoins the original segment. For example, you can see in this picture, okay, there is 1 chromosome over here, and let's say this segment of the chromosome breaks off, and then it will rotate 180 degrees, then rejoin, and this will form a chromosome with an inverted sequence of genes. So how can this occur? Okay, for example, let's say we have this 1 chromosome with the genes A, B, C, D, E, F, and G, H, I, and then this part of the chromosome breaks off, and then it will rotate. It will rotate to 180 degrees, in which the genes D, E, F is now inverted, forming FED sequence. And when this segment joins this chromosome, then we can see a new sequence of genes, which is now ABC, FED, and GHI. Type number 3 will be translocation, in which a region of the chromosome breaks off and joins another non-homologous chromosome. If we look at this diagram over here, we can see that there are two non-homologous chromosomes, chromosome A and chromosome B, and let's say one segment of chromosome E breaks off and joins with chromosome B, and the same goes for chromosome B as well, in which part of the chromosome B breaks off and joins with the chromosome A, and so we will get two types of chromosomes that have changed in their original structures. So this is translocation. Okay, and how can this happen? Okay, again, let's say we have two homologous chromosomes. Okay, it will break off somewhere here, and this part of the chromosome number 1 joins with this chromosome number 2, and as a result we can see a shorter version of chromosome number 1 having only genes A, B and C. Meanwhile, chromosome number 2 will also change in terms of their structure, in which the genes will be having a new sequence D, E, F, W, X, Y and Z. And another type will be the duplication, in which a region of the chromosome duplicates, and so a set of genes will be repeated as seen in this diagram. This segment over here of the chromosome is being duplicated. Thus, we can see a newly formed chromosome with duplicated set of genes. So how can this happen? Let's say we have two homologous chromosomes, okay, in which there will be genes A, B, C, D, E, F, G, H and I. So there are two here, homologous chromosomes, and let's say this part of this gene breaks off, and this segment of this one chromosome will join with this second chromosome which is homologous to the first one, and so we can see a new type of chromosome with a duplicated gene, now having the sequence of A, B, C, D, E, F, G, H, I, and again F, G, H, I. So there will be an additional set of genes for this new type of mutated chromosome.